Thanks everyone. Thanks for joining us this morning and welcome to the May IWN webinar. Today we have the A team from the ARC Training Centre presenting on the topic of transforming biosolids. I'm Michael Thomas, Deputy Director at IWN and R&D Lead at Barwon Water, and I'll be your MC for today's webinar. As always, we begin today's session with an acknowledgement to country, and we acknowledge the traditional owners of the lands and waterways on which we live, work and play, and pay our respects to elders past, present and emerging. Before I hand across to Aravind and the team, just a couple of housekeeping points. First of all, we're already underway and recording. The recording will be available on the Knowledge Hub, so just keep an eye out for the link either in the chat function, but also it'll be distributed in our newsletter and on our subsequent emails. Just a couple of other courtesies that are in play for today. We're obviously just been testing the video, so it would be good or highly recommended if people can just drop their video, except for those that are going to present. So I know we've got teams across Australia today, a number in laboratories. So if those teams that are going to present can have their video on, that would be great, just so we can clearly see who is who in our team zoo, so to speak. Also, just recommend that people do go to mute. Today also, we're using a Menti board for questions. So what I'll do is very shortly, once we get underway, I'll post a link to the Menti chat, to the Menti board in the chat function. It's really easy to use. Just go to the website, punch in the code that I'll put up. And for those that have got a sharp mind, the code is 3366306. That's 3366306. But I'll put that shortly in the chat. Once you go there, follow the links, go to Menti, add your questions into Menti, and you can like and just join the chat function through Menti. So keep a lookout for that. What we'll do is at the end of the presentation, we'll dive into those Menti questions and we'll get the team on board to answer them. So stick around for that one. That's about it, folks. At this point, I'd like to hand across to Aravind Surupaneni. Aravind, for those that don't know, is IWN's Program Manager for Biosolids and Resource Recovery. Aravind, it's over to you to introduce the team.
Thank you, Michael. Good afternoon, everybody. Welcome to this seminar. We're trying something new today. So, as Michael said, the topic is all about biosolids. This is a first time introducing the RMIT team of this ARC Training Center. So what started off with the idea from Kalpit, Professor Kalpit Shah, four years back over a cup of coffee, to start to think about coming up with a biosolids center to service the entire water industry in Australia. We eventually with Professor Andy Ball, as a team, we did it together. And this is a center, as you all know. This is a IWN funded project. The initial center from Victoria, the water industries, people are Western Port water, Western water, Warren water, Colvin water, East Gippsland water, South Gippsland water, Northeast water, Southeast water, Melbourne water. They all joined the center in the funding application. When the center got up, one of our objective was to involve all the rest of the water industry in Victoria. Therefore, we got some funding, a nominal funding from Intelligent Water Networks, so that every water co-op in Victoria are part of the center. Without, I would not steal the thunder, but I think Kalpit is going to introduce the center and the center themes and the logic behind today's webinar format. So yeah, please enjoy. This is something new. Give us a honest feedback. Okay, and over to you, Kalpit. Kalpit, over to you.
I think Kalpit is on mute.
No, yeah. So sorry, Arvind, Andy and I are together. So Andy and I will both introduce. Andy will start first and then I'll take over.
Okay, go for it.
Okay, thank you all very much for joining us today. My name is Andy Ball. I'm the director of the Transforming Biosolids Centre. And with me here is Deputy Director for Academic is Professor Kalpit Shah. We're going to start from a top-down approach today, and I'll give you a very brief overview of the centre and what its mission is, and then pass over to Kalpit, who'll prepare a little more detail regarding today's activities. So the Transforming Biosolids Centre was established in 2020. It's a 5-year centre, and it's part of the Australian Research Council Industry Transformation Training Centres, which are really looking at transforming key Australian technologies in terms of both technical aspects, but also importantly for training the next generation of leaders. And so our overall aim is to transform Australia's biosolids resource. And that's going to occur through innovative technologies, through new knowledge and research, but also to train the next generation of biosolids practitioners. And you're going to hear from some of them today. And we want to guide best practice in the biosolids sector for the future. This is a unique centre. There's no other biosolids-based centre in the world. And so consequently, to achieve this, we've got a whole range of universities, 4 Australian universities, 2 international universities, and over 30 industry partners, all aimed at transforming the biosolids sector in the future. Now, that's all I'm going to say. I'm going to now pass to Kalpit to tell you a little bit about the detail of what the activities are. Kalpit.
So today we have all our centers, postdocs and students are doing 3-minute speeches, more like a speed dating type thing, and they will explain about the research that they're carrying out at RMIT as part of this training center. Training center's main 3 themes are technologies, products and sustainability. So they're all working across these 3 themes. So they will talk about what they're doing, but just to give you a brief idea, on technology side, today we are covering in our presentation technologies like pyrolysis or gasification, more thermal treatment process, then hydrothermal, as well as anaerobic digestion. Then on products, we are looking at blended biosolids application, as well as more transformed biosolids like biochar and their applications. And on sustainability side, RMIT is focusing on contaminants, understanding fate of contaminants. So we'll be talking about ecotoxicology, and also we'll be looking at social acceptance. So whatever these technologies and products we are forming as part of the Biosolids Transformation Training Center, how environmentally sustainable and how socially acceptable those products and technologies are. So today you will hear from all our centers, postdocs and students. So without taking any further time, I'll hand it over to Lydon, who is the coordinator there. So Lydon, it's floor's yours now.
Thank you, Kalpit. Yeah, our first team to speak is the anaerobic digestion team, Chris and Jake. It's over to you. You are spotlighted now.
Thank you, Lydon. Thank you, Andy and Kalpit. So I'm Chris. I'm an industry center postdoc and early career researcher located here at this wastewater lab at RMIT in Bandura, Melbourne. So I personally started working at the Biosolids Training Center last year in August, right after finishing my PhD, together with Tim. Together with Tim, who's online today, not personally here today, and who started his PhD this year, and Jake, who is already in his 3rd year PhD. We are part of a team that works on anaerobic digestion under the theme 1 of the center. And so as Kalpit and Andy just already explained, here at the Biosolids Training Center, we're training ourselves and researching topics that cover various aspects from the pre-processing and processing to the reuse of biosolids, and across those 3 themes, 3 different themes, and they're all somewhat interlinked. But the very first theme is all about improving technologies on the processing side of biosolids. And so Tim and me, we are looking at how to improve the anaerobic digestion process in wastewater treatment plants. And Jake is doing the same just for industrial waste plants. And anaerobic digestion is a very important pre-processing step that determines not only the quality of the actual biosolids that comes out of this process, but also the whole economics of the treatment plants. And that is because anaerobic digestion, it helps degrade that biomass that comes through with the wastewater and the trade waste. It reduces that whole biomass and reduces pathogens, and it also reduces all these organic pollutants that come through as well. All the while also producing biogas for energy in the process. But because anaerobic digestion is a biological process driven by very dynamic and interacting organisms, similar to what actually happens in our gut microbiome, there are some problems that happen throughout the year for operators that they have to manage. And these problems, for example, foaming, and you can actually see that right here, this sludge foaming in front of us, or just generally a low biogas production, they do cost operators money for maintenance, and they reduce efficiencies of those of the biogas recovery. Normally, anaerobic digestion happens in large sealed fermentation vessels where you just basically let the bugs do their thing. And so here you can see anaerobic digestion happen in this column here. This is sludge sampled from a large-scale digester from a Victorian treatment plant. And it is a bit smelly and gooey. We're getting used to it here in our wastewater lab, but we have great facilities here at RMIT, so we're quite lucky that the air is filtered and extracted really efficiently. But so one of the projects that Tim and I are working on here, that we want to help industry to diagnose problems before they actually happen within the microbiome throughout this process happening here of anaerobic digestion to enable more efficient management for operators of wastewater treatment plants.
And as Chris said, I'm working with Andy and Calvert on biogas recovery from industrial trade wastes. We all want to do this by running lab-scale digesters in parallel with full-scale digesters and monitoring the microbiome over a long period of time. We'll find biomarkers that relate to problems and to opportunities. I've already finished initial experimental runs comparing batch biomethane potential for individual effluent streams and trade waste customers. The digesters will be placed in this room and automatically fed with sewage sludge, which we'll get from a full-scale plant, like the Kui-Rup Municipal Wastewater Treatment Plant run by South East Water.
Yeah, so the lab work then will involve extraction and sequencing of DNA, and we also extract metabolites from our lab-scale digesters, which will be set up here right in this room, which we will then manage and monitor here. And then we will develop and use suitable machine learning algorithms to find those really important biomarkers that, as Jake mentioned, relate to problems or opportunities. And once we have those biomarkers, it'll be then possible to integrate them into existing modeling frameworks and then come up with managing strategies to increase the efficiencies for operating anaerobic digestion in Australia. So actually half of this project will be lab work and smelly and gooey stuff, and the other half will be in front of the computer doing bioinformatics and a bit more geeky stuff. So that's us. And now over to Mojtaba and Cameron on the hydrothermal team.
Hello everyone, thank you for the opportunity. My name is Mojtaba. I'm a research assistant at RMIT University, and I'm working with Center on hydrothermal process. And we are here in PC2 Lab at RMIT City campus, and we are trying to develop technologies around hydrothermal processing to improve the existing technology at wastewater treatment plant. And here with me is Cameron.
Hi, I'm Kamrun, a first-year PhD student of RMIT and the University Training Center for Transforming Biosolids. And the outcome of our project will help industry that industry has faced for many years, which is handling the high volume of sewage sludge. And basically, the sewage sludge are the byproduct of wastewater treatment plants.
These wet wastes contain 70 to 99% moisture content, and they carry a significant amount of pathogens. So because of environmental regulations, we cannot just simply incinerate them or landfill them. So we need to think about a more efficient process. Pyrolysis or other thermochemical processes like gasification, they require energy-intensive pre-drying step. And another alternative is anaerobic digestion, which we just heard about, which is time-consuming and also leaves behind the digest state, which is another wet waste stream, which needs to be properly managed. So hydrothermal treatment is our proposed process for handling wet waste streams, because in this process, water at high temperature acts like a solvent. It means that it will become like a non-polar solvent, and it can dissolve biomass, and produce lots of useful products. And also, it will act as a catalyst, because at higher temperature, water gets ionized and can catalyze hydrolysis reaction and decompose big macromolecules. So we can see the benefit of using hydrothermal processing in this, handling wet waste like sewage sludge. And Cameron will help me to explain the reactions.
So we are using our project in this hydrothermal car reactor, the high temperature and high pressure reactor to thermally treat the three kinds of sludge, the primary, secondary, and the digester sludge, and to convert it into a useful product. And this reactor is nothing but a pressure cooker. We just load our sludge in it in a certain water and sludge ratio and turn it on and allow some time to convert it into a value-added product. And here, as a product, we first get the gas, that is mainly the carbon dioxide from here. And the second is the solid residues, that is known as hydrogen, and we can use it as a soil amendment or bioadsorbents. And the second product is separated from the solid residue, that is the liquid portion, and this filtered liquid further goes to the liquid extraction process. And here we got the three product, the bio well with a high heating value, and the second is aqueous that contains a lot of low molecular organics and the ketones. And the most interesting thing is now water industry is concerned about the PFAS, the per and polyalkyl substances, and a hydrothermal treatment can degrade these emerging compounds. And in our project, we are also trying to use two natural catalysts to see whether it will increase the conversion factor of sewage sludge and the product distributions. And as a catalyst, we use the lime and the alum sludge, as these both products we get from the water industry as a wastage material. And after analyzing all of our experiment, we will using the product percentage and the physical and the chemical analysis of these sludge to model a process flow sheet to integrate our hydrothermal process with the existing industrial parameter. And we also trying to do a techno-economic viability assessment that around the sewage sludge of the hydrothermal process. As you know, there is a lot of aspects to explore. We hope our project will help the water industry. Thank you all. And I like to welcome the Starbucks team. They will talk about the pyrolysis process. Thank you again.
Good afternoon, everyone. My name is Sawan Patel. I'm working as a postdoctoral fellow in Biosolids Training Center. In Biosolids Training Center, we mainly focus on the two. My group is mainly focused on two projects, Project 1B and Project 2C. So Project 1B is mainly enhancing resource recovery through thermochemical hydrothermal processes, and Project 2C is mainly focused on future direction of biosolids. So my group is going to talk today on the pyrolysis process. The pyrolysis process is one of the thermochemical conversion process. So pyrolysis converts biosolids to biochar, pyrolysis oil, and the gas in the absence of oxygen. Biochar produced from the pyrolysis process has good quality of physicochemical properties. Based on its properties, we can use biochar in non-agricultural sector. Just say, for example, currently we're working on its application in catalyst as well as in adsorbent. And we successfully demonstrate its application as a catalyst to convert biogas to carbon nanomaterial as well as in hydrogen. And we also demonstrate its application as an adsorbent to adsorb contaminants such as PFAS. And both applications we have successfully demonstrated so far in our lab scale reactor, semi-pilot scale reactor, as well in pilot scale reactor. So now Nimesha is going to explain co-pyrolysis process as well as the lab facility available in our lab.
Thank you, Savan. I'm Nimesha. I'm a third-year PhD student, and my thesis is on co-pyrolysis of biosolids with alum sludge and agricultural waste. Co-pyrolysis is a modified pyrolysis technology, and my focus is on using co-pyrolysis to reduce heavy metal in produced biochar. I also analyzed oil and gas products of co-pyrolysis, and also as part of my PhD, I analyzed the effect of co-pyrolysis on fate of PFAS in biosolids. And to carry out all these objectives, I did many experiments in the experimental setup that you can see today in our background. And today, so I'm going to explain main parts of our experimental setup. I'll start with our reactor. This is our pyrolysis and co-pyrolysis reactor. It is a cast tube reactor. We operate this in a fluidized bed mode, and we use nitrogen as our fluidization gas, which is fed from the bottom of the reactor, and it travels to the distributor plate, fluidizing our sample. And to get it to the required pyrolysis temperature, we put it in our electrical furnace, and then we input the pyrolysis temperature to our controller. And during pyrolysis experiments, gases evolved from the sample are passed through this pipeline, and the condensable gases, which are oil products, is condensed in these condensers, and non-condensable gases are passed through, and we analyze it online from our micro JC. I don't think you can see the screen today, but in our micro JC, we mainly analyze hydrogen, methane, carbon monoxide, carbon dioxide, and nitrogen composition of our pyrolysis gas. And after the experiment, we collect biochar from our reactor and oil from our condensers. I have these samples today. I'm not sure if you can see them. So after the collection, we also analyze these product properties of biochar and oil to further improve and optimize our technology process parameters like temperature and mixing ratio. And there are also many new exciting work going on in our group regarding improving the reactor efficiency. Shivani is working on that. I will let her speak about that.
Thank you, Nimesha. My name is Shivani, and I'm a first-year student of this RMIT University. And as my team has successfully demonstrated the application of fluidized bed technology in producing biochar from biosolids, my work here will be to improve the geometry of the system so that it can give us an improved efficiency. Now, my work will be focusing on adding on the internals inside the system, because as we know, fundamentally, if we add on internals, it will improve the flow dynamics, which will actually increase heat transfer inside the system. So my work will basically be performed using a tool named as computational fluid dynamics, where I'll be trying to improve the geometry of the system so as the efficiency of the system improves by using some numerical method equations. This study cannot be performed on experimental scale before having a fundamental understanding. So my work will basically build up a fundamental understanding on improving the efficiency, which is still pending to be done before industrial application of this process. So we hope to make a significant contribution in this area. And now over to Charles and Cleon to discuss the applications of biochar.
Good afternoon, everyone. My name is Tien. I'm a second-year PhD student, and this is Charles. He's the third-year PhD student. And our work is basically just how do we apply this biochar that's being produced. So we've identified two key areas. For me, it's anaerobic digestion, and for Charles, it's soil cleanup. So in my own research, I try to anaerobically digest chicken manure, which is a huge source of organic waste. And to do that, I mix chicken manure with wastewater sludge. And the goal of this project is to generate biogas, which is the renewable energy. But this process has a lot of instability, mainly because of ammonia buildup. So therefore, that's where the biochar can be applied really effectively because it has absorption capacity. So just to walk you through how I set up my experiment, this little shop motor right here will be connected to a anaerobic digestion rig. You can't really see it very well, so I'm going to lift this up. Yep. And this system is filled with water, and any gas that's being produced will then be collected via the water displacement, and I will then extract the gas using a syringe and then run this through this biogas analyzer, which will tell me how much methane is being produced. In the last project, I've identified really good uses of biochar, and I've produced a lot of methane. There hasn't been any sort of ammonia inhibition. And moving forward, I'd like to apply biosolid biochar. In the last project, I used wood biochar. So I hypothesized that biosolid biochar will also work really well because it's got a lot more inorganic elements, and that's going to help with the ammonium absorption. I'll now pass the time off to Charles to discuss about his soil cleanup project.
Okay, thank you. Man's interaction with the environment took a new dimension after the Industrial Revolution. And one of the adverse effects of our interaction with the environment is soil contamination. And one of the soil contamination that's a problem globally is the problem of petroleum hydrocarbon contamination. And it affects many parts of the world, including developed and developing countries. In my research, I'm trying to see the use of biochar. Biochar produced from Biosolids, if it can enhance the hydrocarbon removal process. And what I do, first of all, I start by, first of all, creating an artificial soil contamination by contaminating the soil with diesel. And after doing that, I try to distribute the soil into pots like this kind of pot you can see here. So when I distribute it, I have a pot that's serving as a control. In that pot, I'm not going to add biochar to that pot. While in another pot, I'm going to add biochar. And when I add it, I'm trying to see the effect, to see if adding that biochar to the soil could help to increase the removal rate, the removal efficiency, or if it will decrease the removal efficiency, or if there's no effect of adding biochar to the soil. So in my previous study, I found out that adding biochar to the soil improved the hydrocarbon removal, especially at the early stage. I found out that adding biochar to the soil led to more than 2 times higher removal compared to the soil without biochar. And I also found out that in my previous study that the treatment condition, the pyrolysis temperature of biochar, the application dose, and also the presence or absence of fertilizer, and also the dose of fertilizer has an effect on the efficacy of hydrocarbon removal. And I found out that based on my previous study, adding fertilizer with the biochar was detrimental to the hydrocarbon removal. In essence, biosolid is enough. There's no need to combine biosolid with fertilizer in order to improve the hydrocarbon efficiency. And in my next study, I'll be trying now to look at the mechanism, trying to find out the mechanism. How does biochar affect hydrocarbon removal? And the good thing about our study appears to be one of the first study in Australia to apply biochar in the cleanup of contaminated soil. Outside Australia, there have been many studies carried out to use biochar to clean up contaminated soil. And overall, my project and this project is like a win-win solution in the sense that we are trying to see how we can use a waste product, biosolid, if we can use it to solve other environmental problems like soil contamination and the problem with anaerobic digestion. Thank you.
All right.
So Ibrahim, over to you.
All right, thank you, Tien and Charles. My name is Ibrahim Akin. I'm a third year PhD student at RMIT University. I'm also working with the Biosolids Center. So I'm working under the team of improving product application and enhancing sustainability. As we all know, Biosolids is a final product obtained from the wastewater treatment process. About 70% of Australian-produced biosolids are being applied to agricultural soils. However, there are issues with contaminants. The contaminants in biosolids can be classified into three groups: organic contaminants such as PFAS, inorganic contaminants such as heavy metals, and microbial contaminants. In my own work, I'm trying to explore treatment of biosolids towards reducing the level of inorganic contaminants, mainly heavy metals. And to do this, I'm going to be using acid extraction process to remove the amount of heavy metals in biosolids. So this morning, I'm working on removing heavy metals in biosolids due to acid extraction process. So I have 3% sulfuric acid, very dilute acid solution. I'm going to mix that with my biosolids, and I'm going to subject that to room conditions of heating and also stirring. After 30 minutes of extraction, the mixture will be separated into liquid phase and solid phase. The solid phase is a treated biosolids, which, based on my studies, I found that up to 80 to 90% of persistent heavy metals in biosolids can be removed, and the final treated samples can be safely applied in agricultural soils. So far, I'm trying to optimize the process further to see possible scale up for wastewater industries to integrate that with their existing treatment facilities. I'm now going to pass on to my colleague, who is still going to talk about pathogens removal in biosolids. Up to you, KJ.
KJ, if you can share your screen and play the video, will be great.
Hello, everyone. My name is Kraivut Jansipiboon. I belong to Project 2A, which is Understanding and Improving of Fertilizer. My project aim can be summarized into two keywords: pathogens and antibiotic-resistant genes. I want to study survival of pathogens from the biosolid fertilizer in agricultural application. Also, I want to study antibiotic-resistant genes, persistent and horizontal gene transfer to local soil microbes. Currently, the biosolid in Australia, even after thorough stabilization after a long period of time, it still contains pathogens and possibly antibiotic-resistant genes. The pathogens can potentially contaminate soils, plants, and then pass through farm animals and eventually humans. My project focuses mainly on an interaction between microbes from biosolid and microbes in the soil. The antibiotic-resistant genes may exist as naked DNA outside the cell or inside alive cells. The technique I'm going to use is called viable PCR. It is better than conventional PCR that can only tell total DNA but cannot differentiate which one is live or dead. Soils have been reported in many cases to contain dead DNA in large amounts, which could go up to 40% of total DNA. This means that the conventional microbial community analysis could likely be inaccurate because of the inflation from the dead DNA. However, in some other cases, the dead DNA did not cause significant bias. This is mainly because soils are very varying in nature. The viable PCR can also differentiate whether antibiotic-resistant genes are inside live cells or being naked outside the cells. Another important point is that most of the studies using viable PCR on soils were done in natural habitats, which are greatly different from agricultural environments, especially agricultural soils implemented with biosolid fertilizer, which has a lot of microbial activities in there. This would yield very interesting results. Behind me is MySeq. This machine will be used for sequencing DNA from microbial community samples. I will use this along with digital PCR for complete analysis. I hope this presentation is easy to follow for you and give you a main idea of my project.
Perfect. Thank you so much, KJ. So after what we listened for all the technologies and application of biosolids and biosolid blended and transformed materials or products, it's very important to make sure those products are risk-free and safe before we apply to the environment. This is Leiden Kudur I'm a postdoc in the center of the ARC Biosolids Center, and I'm working on a couple of projects across the 3 themes. So I'm assessing the ecotoxicity of the biosolids products and transformants to make sure they, as I said, they're risk-free. So we're assessing the improving technologies in theme 1 and the enhanced product application in theme 2, and make sure those meet all the requirements, all the objectives of theme 3, which are ensuring the sustainability, and also to develop a risk-based framework for the quality of biosolids products. So what I'm using here at the moment is a called microtox test or toxicity test. This is a standard wastewater and effluent toxicity test. It basically uses a marine bacteria that is a bioluminescent bacteria, which means it produces light as part of the metabolic activity. But when this bacteria get exposed to any of chemical that is toxic or toxic for their metabolism, the light emission will reduces. So this machine here, it's called Microtox Analyzer. This is a module 500. Basically measure the light emission by the bacteria after it gets exposed to different concentration of the chemicals. And we record all the data from here. We put it in the associated like software in the computer, where will give us the endpoint of the test. At this test, the endpoint is what we call the effective concentration 50 or EC50 value, which is defined as a concentration of contaminant that basically affects the light emission of the bacteria in this case by 50%. And that the endpoint is a very standard endpoint of the toxicity test. But this is not the only test we use in terms of ecotoxicity, but we also use the earthworm toxicity test for any soil application. Again, the endpoint of that will be a bit different from this one. In earthworm, we will look at the lethal concentration 50, because we look at the concentration that kills 50% of the test population. So again, this ecotoxicity test is just, we run it across all the projects and across all the products, just to make sure that all the technologies we are using and the products we are developing from the biosolids are safe and risk-free before we apply to the environment. That's all for me. Thank you for now. I'll just hand it over to Patrick, who will just take you through the social concept of biosolids. Thank you for now.
Thank you, Leiden. So yeah, my name is Pat Bonney. I'm an environmental social scientist at RMIT University and industry postdoc at the ARC Training Centre. I'm representing today Project 3D of Theme 3, which, as Leiden mentioned, is about ensuring sustainability. And the key issue that Theme 3 is addressing is the following. You know, all these fantastic and innovative technologies and applications for biosolids in creating these new opportunities for water, energy and the agricultural sectors will ultimately be difficult to implement if environmental regulations, if policy frameworks and public perceptions are out of step with this science. So Theme 3 is considering various topics that are central to the improved management of biosolids. It includes developing vertical classification systems, national guidelines and frameworks for biosolids risk assessment, as well as measuring public perception and community engagement practices. Now, in my project, Project 3D, and with my colleagues from RMIT, the University of New South Wales and University of Western Australia, we're focused on this last issue around stakeholder engagement and social acceptance. Our core aims for this project are to conduct research on the social and economic dimensions of biosolids, including the factors that are shaping its uptake and scaling. We're also keen to help identify some practical approaches to enhance biosolids adoption for its products and technologies. And further to this, we're hoping to improve the knowledge and capacities of researchers and water utilities as they work towards improved engagement with end users and public. So we've begun our first phase of our research, which involves reviewing existing literature and plan to conduct interviews with key people in the biosolids industry to understand the current state of stakeholder acceptance and engagement in Australia and how it's currently sort of understood and practiced. And so following this, we'll be conducting some broad scale surveys to investigate these issues, such as on the factors that are shaping biosolids management, and with a particular emphasis on its market acceptance. And this is led by my colleague, Dr. James Hayes from the University of New South Wales. And finally, we aim to conduct some research on 2 biosolids value chains to generate some really in-depth understandings of what it actually takes to transform biosolids into a resource of value to farmers and other potential end users. Our preliminary findings have really highlighted that stakeholder acceptance of biosolids products is really not a straightforward process, which is something that I'm sure you're all aware of. And this does align with almost 3 decades of social science research that has consistently shown that end users have a wide range of valid reasons for not adopting these logical new innovations, including competing demands and priorities, as well as some of the transaction and opportunity costs that this adoption could entail for them. So Project 3D and Theme 3, we look forward to exploring these issues further and hopefully delivering some of the empirical and strategic knowledge that's going to be necessary to ensure that the ARC Training Centre's research is genuinely impactful. We're also currently searching for some suitable case studies to inform our research for this value chain analysis. So please do get in touch if you'd like to learn a little bit more about this or would like to get involved. I'll now pass it back to Leiden, I believe, who will take us through the Q&A. Thank you.
Thank you, Patrick. I think it's Andy and Calpit who are gonna run the Q&A session. So I'll just probably, you don't have your camera on, oh yeah, you do have it now. I'll just spotlight you, Andy and Calpit. So for any Q&A session.
Thank you very much, Leiden. We've got some really good feedback and questions from the audience, and I guess I've just put them into the top questions. And the first one, if Jake is still with us, I'm sure he is. There was a question which was supported by a number of people about what kind of trade waste customers are you looking at in terms of your anaerobic digestion work?
Yep, sorry. Thanks, Andy. I've been really lucky to work with Greater Western Water. They've been very generous with their data and their time. So I'm looking at a very wide range of trade waste customers in that Greater Western Water catchment. So it can range from sites that do about 10 meg a year of wastewater through to 600 plus megalitres per year. Looking at anywhere from food manufacturers through logistics companies and chemical producers. And yet looking at their final pre-treated waste as it hits the sewer, but also some of their effluent streams before they hit their DAFs or pH correction.
Thank you very much, Jake. That's a good answer to the question. Going down to the next question, in fact, was an observation, I think, about from Boron Water and the research that's happening through Deakin looking at alum sludge and ability to absorb PFAS, getting some interesting results. I don't know, Calvert, do you want to say anything?
Yeah, no, it's good. Good to know. I think we have heard this also from other water industry colleagues that Dickin and Barwin's working on looking at alum sludge potential for PFAS absorption. What we're doing is using alum sludge into various thermal and hydrothermal processing as a co-feed. And the idea there is to reduce the resultant transform products like hydrochar or biochar, the heavy metal concentration, but also get some synergistic effects like having the contaminants being destructed, but also the yields of the final products can be improved and all. But now it would be great to do a collaboration with Dickin on that. And I think some of you might know that ND and I now recently been funded a state government grant to work with Dickin on a circular economy hub type project. So we are actively working with some groups in Dickin, but please give us a feedback who we should be contacting in Dickin to do this type of work in more collaborative way.
Excellent. Thank you, Calvert. The next question is about the observation that we've got lots of research, lots of great ideas, but I think the question is, has any research been done on what the highest value of these options might be? And I think the answer we heard in our project 3D that from Patrick is yes, that's part of the consideration. And I think one observation that we can already make, and I'm sure everybody is aware, is that there's no one solution for all of the answer to what we're going to do with biosolids in the future. But there'll be a lot of different solutions, and techno-economical analyses are an important part of all of these this research areas, and all of the postdocs and PhD students on their particular technology will be undertaking that analysis. And then over in theme 3, there'll be a lot more critical analysis together with risk analysis of those outcomes. So yes, it's a really important part of what we want to do. And this is a 5-year centre. A lot of these projects, we're formulating ideas, we're making connections, we're already thinking of how we continue the research and go beyond those 5 years. So we hope all of our partners, many of you out there, will continue the journey with us, and we'll be providing more information on outcomes and next steps. So yeah, very much part of what we're doing. What kind of agricultural waste has been co-paralysed with wastewater sludge and alum sludge?
Nimesha, do you want to answer this? So the question is, what kind of agricultural waste you have selected for co-pyrolysis, and how did you select them?
So I have selected wheat straw and canola oil straw for my PhD work mainly, and I did this based on a mapping study of Victoria. And to select these two types of waste, I considered several factors like the volume generation and how they are distributed based with respect to the wastewater treatment plants, and also physiochemical properties like heavy metals and CHNS. And based on that, we selected two waste products, wheat straw and canola oil straw.
So
those are
resource mapping. So we cross Victoria and water utilities to find out which is available close to wastewater treatment facilities in Victoria. Okay.
Yeah. And the high what is the volume generation, whether it is generated in a high volume or not.
Yeah. No, thank you.
Absolutely. There's a question I'm just going back up to in our top questions. Keen to see further work to build knowledge and evidence on the carbon sequestration, emissions reduction benefits of biochar production and use. Do you have any plans on this topic? Again, maybe perhaps being part of theme 3, Patrick, you might have some insights into that for us.
Thanks, Andy. I'm just looking for the question now. carbon sequestration and emissions reduction benefits. Sure, this is definitely something that is inside the scope of Theme 3. It's not necessarily something that we're going to be focusing on Project 3D, but for sure, this has been a conversation that we've been having around the benefits of biochar for carbon sequestration and emissions reduction from biochar production. So yeah, we can definitely keep up to the industry up to date with the work that's going on in that area.
Thank you, Patrick. There's still a couple of questions. I'm conscious of time, but we'll try and to get through these questions. The next question I'm going to ask Tien to prepare an answer for, and that is, do you believe the methane produced via chicken manure and wastewater mixture is sufficient enough to produce electricity if upscaled? Can it be commercialized? Tien.
Hi, thank you for the question. Yes, I believe it can be commercialized. On a bigger scale, it's really possible because in my last project, I got up to about, the high was about 82% methane as a total volume of gas produced. And from a mere 20 grams of chicken manure that I used, I produced about 1,300 mils of methane over the course of about 40 days. So I think if we can upscale that, there's no question that it can be commercialized. I really believe that it can be a product of very high value. It's just so much chicken manure sitting around. I think it's such a waste to just, compost them or just to throw them away. So yeah, I definitely believe in it. That it can be commercialized.
Thank you, Tien. Very encouraging and great work. I think the final question I have is about lots of efforts on new treatment methods, but have we quantified the benefits of land application, the benefits on carbon sequestration, etc.? There's a lot of work going on in Theme 2 for that, and that's actually led mainly by University of Western Australia, who are carrying out a lot of that work. But I don't know if Charles or perhaps even Christian, you may have some thoughts on the impacts of the treatment methods on the benefits of the land application. Charles.
Okay, thank you for the opportunity. So for the land application in the aspect of soil remediation, because normally when a pollution exercise happens on the ground, happen on the soil, so they use different methods like maybe excavation, they use land filling and other methods to clean up the contaminated soil. And sometimes they even allow it to remediate on itself biologically. But adding the biochar now to the soil now will now speed up the process, will now increase the efficiency. And adding biochar to the soil is a way of even returning carbon to the soil. It can help in carbon sequestration. Okay. Yeah, I'm done.
Thank you, Charles. That's good. That's, as far as I'm aware, all of the questions, unless I've missed a couple. But if not, in the interest of time, perhaps back to Aravind to wrap up or to finalize. Aravind.
Sorry, I'm sharing my screen.
Yeah.
Folks, what a fantastic presentation. Something unprecedented. We never did try this model before. Great teamwork for water industry colleagues. We've been practicing this for the last four weeks. I missed the last Friday's presentation, but it's great teamwork across the RMIT contingency. Look, we fantastic teamwork. Okay, everybody there, good work. RMIT team. I think we want to replicate this across our other three university partners in University of in Queensland, University of Queensland, University of Western Australia, and University of New South Wales. And by the way, it's a spoiler for you. September 5th, 6th, and 7th. There's going to be annual symposium in Sydney at University of New South Wales. This is the Biosolids Training Center annual symposium. There will be a 2-hour session on sponsored session from Australia New Zealand Biosolids partnership, ANZBP, and the third day there might be a field trip from Sydney Water. But I think the first 2 days are very crucial for the industry partners. Thank you. Damien is there. By the way, Damien is one of our chief investigators from University of Queensland on team 1. Okay. I don't know whether who else? Anybody there from our chief investigators? Stuart is there? I don't think he's there. Anyway, I really want to, in the interest of time, it's almost 2 minutes before 1 o'clock. Michael, I'll hand it to you, Michael.
Yeah, thanks, Aravind. Well done, team. Aravind, Andy, Calpert, to all the presenters out there at the ARC Training Centre. You know, imagine trying to do this without technology. I mean, this would have been, you know, just so difficult to do. So just great to see this level of fundamental research happening on such an important issue for the water sector, and you know, and other industries as well. So you know, just very grateful from the IWN team and for everyone here today for all the effort that has gone into it. And also great to see that the white lab coats are still in fashion around the trap. So that's super encouraging as well. So from all of us, I'd like to give a virtual round of applause for the team. And again, very grateful for your efforts, team. Thank you. I'd also like to just do a quick shout out to all the participants for the questions, for joining in, for coming along for the journey today as well. A shout out to the knowledge sharing team. Alyssa does a lot of behind the scenes works just to pull the webinars together. A quick reminder as well that our next webinar will be on Thursday, the 16th of June. So just stick around or look out for details on that one. That'll come out in the IWN newsletter. And also don't forget to visit the Knowledge Hub. If you're not signed up, the link's in the chat. We also put in the Transformation Centre website as well. So heaps of information going on there. If you want to get in touch and learn more about it, Aravind breathes this night and day. So make sure to get in touch with Aravind or through the team at IWN. That's it from ourselves. Thanks very much for joining in and have a great day. See you, everyone.